Hi guys, welcome to the second part for chapter 16. In this chapter we're going to look at dihybrid crosses and the ins and outs and sort of how they work. Okay, so let's jump into it. Okay, dihybrid crosses. Now, before we were looking at monohybrid and monogenic traits where you only have one gene involved. A dihybrid cross is when you now have two genes involved. Yeah, if you want to look at the crosses that occur between individuals exhibiting two particular traits. Okay, so let's go back to our good friend Gregor Mendel and his peas and use his peas as our prime example. Let's pick two genes, not one trait but two traits, and look at how the traits are now interacting with one another in total. Okay, if you have let's say the pod color of a particular pea pod is determined by the letter G, where a capital G and lowercase g, and pod shape determined by capital F, lowercase f. Okay. Now the alleles that are there, if the pea plant has a capital G, it means it has green pods. If it has a lowercase g, it will produce yellow pods, indicating that yellow pods is a recessive trait. If it has capital F, it is firm pods, and if it has lowercase f, it has wrinkled pods. Once again, lowercase f, wrinkled being the recessive trait there. Okay. Now, in example 1, let's say you pluck a particular pea plant and it has this particular genotype of capital G, lowercase g, which means it is heterozygous for pea pods, green pods, and capital F, lowercase f for firm pods. Okay. Now, the phenotype you would then expect, if this was a complete dominance trait, to be green, firm pods. Okay, the pods are green and they are firm. Okay, and what that means is that particular individual pea plant will produce pollen or ovum that will have the gametes of the following genotypes. Okay, it will have capital G, capital F, capital G, lowercase f, lowercase g, capital F, lowercase g, capital F, because that is all the possible combinations of mixtures that will occur within the pollen or the ovule. Yeah, so you got that one there, you've got that one with that one over there, you've got that one with that one over there, and that one with that one over there. Yeah, resulting in 4 different possible genotypes. Okay, let's go with the second example. Let's say, for example, you have a plant with yellow pods that are firm. So yellow being 2 lowercase g's, complete dominance, but in this case a recessive condition, but still has the firm pods, right? Because both of those alleles are lowercase g, all the pollen and the ovum produce are going to produce lowercase g's, and then you've got your even capital F to lowercase f ratio that's being there. So depending on that, you've now got not 2 possibilities on your Punnett square, but you've got 4 possibilities on your Punnett squares. Okay. Let's pick one of them. So we're going to pick capital G, lowercase g, capital F, lowercase f for both individuals, and you mix 2 together, you are going to get a very big Punnett square. Okay, showing all the possible outcomes of the offspring of these 2. So you can see there, in your original Punnett square, the alleles are represented by only 1 letter, but in this particular case, because it is dihybrid, it is 2 genes. So now I've got the G allele and the F allele and all the possible combinations that are there for a heterozygous individual on one side and all the possible combination for another heterozygous individual on the other side, right? You can see some of the common traits coming through. Because yellow pods are recessive, you've still got a 1/4 chance of having yellow pods, and then because wrinkled pods are a recessive condition, you also have a 1/4 chance. 1 there, 1 there, 1 there, and 1 there, and the rest being most likely to be green and most likely to be firm. Yeah, but once again, the numbers are slightly different. Again, yeah, where you can have 1/16 of a chance of a yellow wrinkled pod, and then 3/16 of a chance of having a yellow firm pod, 3/16 of a chance of having a green wrinkled pod, and you've got 9 out of 16 chance of having green firm pods, yeah, because that's the highest possibility that's being presented there. Okay, so that is what a dihybrid cross looks like, and when you cross them, you have to be able to list both the alleles of both genes along one side and along the other, and then identify what the possibilities or the outcomes are for that. Okay. Now, dihybrid crosses are a little bit more complex, okay, because in monohybrid crosses, you've only got one gene on one particular chromosome. Because you've got now two genes, you have to determine whether they are linked or unlinked, okay? Unlinked genes is when they are on two different chromosomes, and therefore, when they're being sorted through the process of meiosis, they end up on different chromosomes, and they are unrelated to one another. The example of that being majority of Mendel's Ps are unlinked genes, and we denote it with a semicolon between the gene alleles. Okay, so in the case of Mendel's Ps, there with the green and the firm pods, the semicolon separates the G genes, or the green pod and yellow pod gene, and the firm and wrinkled pod gene. Okay, if, however, both of the genes are found on the same chromosome during that meiosis phase, when they split up, they are most likely to end up on the same chromosome again, and so they are linked when we see that they're on the same chromosome, and there is only a very small chance that they're going to be separated, and that's usually through recombination. Okay, and when that happens, we don't use a semicolon, we denote it with a slash or a dash. Okay, so for example, if you have capital A lowercase b, dash lowercase a capital B, representing them on the different locations but on the same chromosome. Okay, linked genes affects the chances of the alleles of being passed on together. Yeah, if they're linked, they're more likely to pass on together than apart, and the outcome percentages are going to be different again. Okay, what is that going to look like? So in this particular case here, let's take a look at if you have genes A and B occurring on different chromosomes. Yeah, here are your homologous pairs, and remember, they are now an X double-stranded because of the DNA synthesis. And then what happens is these ones get sorted into the sister chromatids, and then what happens is during the meiosis phase, they all get split up into different gametes, yeah, whether it be sperm or egg. So this particular gamete has capital A, capital B. This one here has capital A, lowercase b. This one here has lowercase a, capital B, and this one here has lowercase a, lowercase b. And so you've got four different gametes. Not only do you have four different types of gametes, you have four different gametes that are occurring of the same probability or possibility. Yeah, the chances of each of these ones is 1 in 4 occurring for the gametes. However, if genes A and B are found on the same chromosome during that split up phase, right, you're only going to get two possibilities. You either get A and B capital or A and B lowercase moving into each of the gametes. So there's only two kinds of gametes produced here. Now, this particular scenario is not including recombination, yeah, which we'll look at in a second, but you can see the difference there. Yeah, 4 kinds of gametes in equal ratios, 2 kinds of gametes if it is complete and there is no recombination occurring, and if there is, it's going to be a very small percentage chance. Okay, so let's go with an example. So rhesus blood type and elliptical blood cells. Okay, the RHD gene determines rhesus blood type. Rhesus is a particular type of antigen similar to ABO, and that sits on the blood cells. If you have a capital D allele, you have rhesus positive blood. Yeah, you have the rhesus antigen. If you have a lowercase d allele, you don't have the rhesus antigen at all, and you therefore have rhesus negative blood. Okay, a second gene, the EPB41 gene, determines the shape of the cell. If you have a capital E, it has elliptical, which is this kind of oddly crooked shape, and if you have a lowercase e, you have normal blood cells. Okay, so here is a diagram to show what it looks like. Yeah, rhesus positive shows the D protein antigen sitting on the blood cells, and rhesus negative having no antibodies, and that showing your normal blood cells versus elliptical. Okay. Now, both RHD and EBB41 are located on chromosome 1, and therefore they are linked. Okay. This means that during meiosis, there is a small chance that they will be separated into the alleles, but the majority of the time they are going to land on the same chromosome. Okay. So let's take a look at that. Yeah. So if we assume that Sarah is heterozygous, and she is heterozygous for both traits, right? Remember, they are linked, and so we use a dash to or a slash to separate them. Yeah. If she is heterozygous for both traits, she is capital D, lowercase d, capital E, lowercase e. Yeah. And these are her possible gamete combinations. Capital D, capital E is one possible gamete, lowercase d and lowercase e for the other gamete, right? And there's a small chance that that's going to cross over to capital D, lowercase e, or lowercase e, capital E, lowercase d. But for the most part, that is going to end up in one gamete, and that is going to end up in the other gamete. Okay. Now, Dave, on the other hand, is homozygous for both traits, which means, homozygous recessive for both traits, which means he only can produce one type of gamete, which is lowercase d, lowercase e. Yeah. So we use that to show that it's linked because they're on the same chromosome. However, he only produces one type of gamete. It doesn't matter what combination he does; he's only going to produce one type, right? And so that's what it looks like, right? So for the most part, because they're linked, when they split up, that's going to end up in one. That's going to end up in one, and there's a very small chance that that's going to cross over to that side, and that's going to cross over to that side. Okay. Now, Dave, on the other hand, because he's homozygous recessive for both traits, it doesn't matter what combination he has. He will always produce only one type of sperm, one with a lowercase d, lowercase e, in all scenarios. And here are the ratios. Okay. You can see the 0.49 represents its percentage. 0.49 referring to 49% chance, 0.01 referring to a 1% chance there, okay? So Sarah is most likely to produce capital D, capital E, and lowercase d, lowercase e eggs, but there is still a 0.01%, 1% chance that she's going to produce recombinant capital D, lowercase e, or 1% chance of lowercase d, capital E, right? Now, if that happens, we say that these eggs here are parental or non-crossover, and they have a higher possibility. The parental and non-crossover eggs always have a higher possibility than the other one, which are your recombinant eggs, because recombination is a very small percentage chance of separation, and so there is only a 1% chance of separation for those ones there, but a 49% chance for these ones over here each, right? And so based on that cross, you can determine whether or not their kids are going to have rhesus positive or elliptical blood, or rhesus negative or normal blood, right? So in this particular case, they're most likely to have rhesus positive but elliptical, or rhesus negative and normal blood. Yeah, they have one condition or the other. There is a very small chance they're going to have rhesus positive and normal, and one very small chance of being rhesus negative and elliptical. Okay, this is going to take a bit of time to get used to, so it's worth just thinking about it and considering it. Okay, we're going to take a look at test crosses for dihybrids now, which are a little bit different from monohybrids. Okay, so in monohybrid, we were using it to identify whether an individual is homozygous or heterozygous. However, in a dihybrid crosses, we are using it to determine whether the genes are linked or not, and how closely linked these genes actually are. So a dihybrid test cross is a test cross where you mix a double homozygous recessive for both traits. Yeah, now that's a bit of a mouthful, but basically it means all lowercase letters, okay? And a double heterozygous genotype, which is the perfect mixture of all combination of capitals and lower cases. Yeah. Now the reason why we picked these two specifically is because we then understand that because of the double homozygous recessive, the first parent can only produce lowercase for both traits in the gametes. The second parent will produce every possible combination of gametes, yeah? And so what we look at then is the offspring ratio. If the offspring ratio is 1 to 1 to 1 to 1 for all the possible combinations, then we know that the genes are not linked. Because if they are in equal ratio, then the genes were not on the same chromosomes, and therefore they have an equal chance of being split up into each of the gametes during meiosis. Okay. However, if the ratio isn't 1 to 1 to 1 to 1, we know that some of the genes are linked and are occurring on the same chromosomes. Okay, or the two genes are linked. So coming back to our guinea pig example, right? If you cross a guinea pig with lowercase bf and you cross it with a heterozygous individual for both gametes, right? And you end up with a ratio of long black, long white, short white at a 1 to 1 to 1 ratio, then we know that it is therefore unlinked. However, if, for example, we see more of a likelihood of long blacks and short whites, but very few short blacks and long whites, we know that the genes are linked. Okay, and that's what the test cross is all about. Okay, we can also use the test cross to determine how closely linked these genes are. You can figure out exactly, map it out, where along the line it is based on the chances of the offspring. Okay, so the distance between what we call the gene loci meaning location, the gene location on the chromosome can be determined by using the formula 100 times the number of recombinant offspring. There should be a B right there. Okay, recombinant offspring divided by the total number of offspring. Okay, so let's go with our example from class, and we're going to look at Shaggy and Honeypots, which is the 18 guinea piglets, right? Let's say we test cross it. Yeah, Honeypots being a short white guinea pig, and she can only produce one type of gamete, BF lowercase. Yeah, Shaggy containing is a heterozygous and will have all 4 possible combinations there, right? Now let's say they interbreed and they cross, and you have these ratios. You have 2 guinea pig offsprings that are long black, 2 that are short black, 2 that are long white, and 7 that are short white. Sorry, I think I was meant to say 7 over here as well. 7 long blacks, 7 short whites, 2 short blacks, and 2 long whites. Okay, and so what you've gotten here is an uneven ratio, which means immediately you can tell that it is linked. Okay, not only can you tell that it is linked, you can determine exactly how much of the linkage is there. Okay, so what we can do is we can count up the number of recombinant offspring. There's 2 here and there's 2 here. How do I know that's recombinant? Because they are the lower ratios. Lower ratios are always the recombinant, because recombination has a smaller chance than your parental non-crossover ones. Okay, so I go 4 times by 100 divided by the total number of offspring that I produce. Yeah, 7 plus 2 is 9, plus 2 plus 7 is 18. So 100 times 4 is 400 divided by 18. Okay, and that comes to whatever the number that is. Okay, and so that actually tells you an estimation of the gene loci distance, or the distance between them. Okay, so we can conclude there that the genes are linked and the genotypes can be written in that kind of fashion. Right, now here's another example. Here the gene loci is a little bit lower. Yeah, you can see 44% chance of parentals, 6% chance of recombinant, which indicates there that there is actually 12 map units that separate between the 2 genes. So in this particular case, E is located up here, and it is actually separated by F, which is 12 map units apart, which is the total number of recombinant percentages. Yeah, so these map units, you can actually figure out how far apart they are on that. Now the word map unit doesn't really indicate anything. It just is a generic term for separating an arbitrary distance between genes. Okay, it doesn't really mean much, so to say, but it's just useful to understand how far they are from one another. Okay, now we'll finish up on this looking a little bit at genetic screening and testing and what they are. So a genetic test is when you test a person's DNA to identify any particular differences in the genetics for diseases and abnormalities. Okay, when people go for a test, they look at the genetics and they look for abnormalities within the genome. Okay, A genetic screening is when you test a whole population for a particular disorder. Yeah, so you screen it. You want to see and you want to inspect every individual for a particular disorder. Yeah, some of the benefits of this. It's important to screen and test for certain genetic conditions because early detection is beneficial. It helps identify those diseases, and then it can lead to effective treatment or management. If parents can figure out whether or not their child has PKU from birth, they can then manage their diet and figure out ways of minimizing the effects of phenylketonuria. Yeah. Now, there are some downsides. So obviously, there are a few breaches towards looking at personal information and a breach of privacy of the individual. When we take their genetic information, we screen it, and then we store it in a data bank somewhere. That's personal information that we're storing, and for some individuals, it makes them uncomfortable to know that their genetics are being stored somewhere. Okay. So in Australia, we do this thing called newborn screening. Well, actually, we do it everywhere around the world, but in Australia, we have newborn screening, and the babies are screened shortly after birth, looking for specifically a few inherited disorders. So PKU is one of the examples, 1 in every 10,000 babies. Cystic fibrosis is also screened at the start as well, 1 every 2,500 babies. Galactosemia, so the ability or the inability, to break down galactose, which then builds up in the blood, is 1 in 40,000. And then also hypothyroidism, when your thyroid gland doesn't function properly and you need to be treated with hormone tablets, occurs in 1 in every 3,500 babies. Okay. Within the first few days of birth, usually what happens is a blood sample is taken from the baby's heel, and then the sample is then analyzed using mass spectrometry, which then looks at the levels of other amino acids in the blood, indicating for particular diseases. So if the level of phenylalanine is super high, then we can see that the individual might be suffering from PKU because they can't break down that particular amino acid. Okay, here's the flow chart. I won't spend too much time on this, but the basic idea is that for medical practitioners and doctors and surgeons and all those people, right? When a new baby is born, you need to get consent from the parents to screen the baby first. Okay, if the agreement is not given, the concerns are discussed with the parents and the benefits of it. If the parents still don't want it, then the test isn't performed. But if the parents agree, or if they agree in the first place, you take the sample. Once you perform the test, there are a few different sort of methods. If nothing comes up, you store the card for a later day. If something does come up, you confirm the test. You still store the card, but you now move in towards the management side of things. Okay. And so the idea is that it is helpful to determine what the action plans are for individuals that are born with particular diseases or abnormalities. Okay. Here's a diagram showing what happens in mass spectrometry. Mass spectrometry. I keep saying the word wrong. But here is a particular chart to show the frequency of different amino acids. Yeah, you can see alanine's there, leucine's there. And the other ones are all there as well. PHE, phenylalanine is there, and you can see that in an individual with PKU, in this particular diagram, that is a big spike, right, which indicates that this individual is suffering from the inability of breaking down phenylalanine, and then it's building up in their body, yeah, which is what that indicates. And then you move into effective management strategies. Okay. So genetic testing, on the other hand, can be carried out for various reasons. Yeah, now genetic testing is up to the individual. Yeah, patients' family history is often first looked at, and then you put counseling, and then you then perform the genetic test. Now, there's a few reasons for that. Breast cancer, so BRCA1 and BRCA2 genes can give an indication of high risk. Now, they are not the only indicators, and they are by no means the final indicator either, but there is an association between them. Okay. The second one is Huntington's disease, the HD allele. The signs usually appear to mid to late 30s, but then when that happens, you can get yourself tested to check to see if you have Huntington's or not. Okay. Now, when an individual goes in for the testing before they exhibit any of the symptoms, we call that pre-symptomatic testing. Yeah, they're basically preempting. They're anticipating the particular condition, especially if it appears in their family history, and they're encouraged to be to be screened, to be tested, so that it can be managed and you can take the action plan accordingly. Okay. There's a few other reasons as well. So genetic testing also looks at paternity, so who the father is in individuals. It can also be used to diagnose suspected inherited conditions, especially if you don't know if it doesn't occur within the family history. And it also is useful for guiding medical treatment as well. Okay. And the last one is genetic testing is also particularly useful for identifying carriers for particular conditions. Okay. Oh, and also for forensic purposes, which is sort of for police and investigation purposes behind genetics and stuff like that, and matching criminals to crime scenes, etc., etc. Okay. Okay, so we're going to finish it there. Next lesson, we're going to look at family pedigrees. I hope you guys enjoyed that, and I'll catch you guys then. Okay, bye.